Then we'll go ahead and just pick up where we left off Monday when we were last lecturing. Last thing we were getting ready or got to talk about was the different classification. Really, this is the only thing we got to talk about was the classification of sensory receptors. Talked about the way that we classify them structurally, looking at them on a microscope based on their location and then what they detect. And so that brings us now to really get to talk about what they do, how they work, different types, and that kind of stuff. Which is what we're going to really spend the rest of the lecture talking about. Yeah. Regardless of the type of receptor, they all perform a pretty cool process called encoding. Okay, I'll let you guys write out if there's anything to write out. Nothing to write out. Okay, so this basically allows you to detect whether something's a light touch versus a firm touch. And it has everything to do with the stimulus strength, the strength of the stimulus. And that controls how frequently we're sending in action potentials to our brain or spinal cord. So let me read through. How can your sensory systems detect stimuli of differing intensity if all nerve impulses are the same size? Remember, an action potential, as long as we hit threshold, is going to end up the same way. Running through depolarization to positive 30, 35, somewhere in that neighborhood, dropping back down by repolarization to negative 70. And regardless of if it is a real strong stimulus or a weak stimulus, if we hit threshold, it's the same thing. Okay. But the frequency in which we transmit action potentials will be different. Okay. And that is what we can show here on our bottom left figure. Okay. We've got a singular unipolar neuron. Okay. On the left side of our graph, we're using a very weak stimulus, and we are completely stimulating it from the point where we hit the on to the off. Okay. So maybe we're just pushing in really weak and barely stimulating this sensory receptor, which causes just a very weak generator potential to be created, which ultimately causes a burst of action potentials at the beginning, but then they start to progressively widen out. So that ultimately ends up with less action potentials transmitted inward to the central nervous system. Versus our strong stimulus here on the right, which creates a much stronger receptor potential, ultimately causing a lot more action potentials and them to be located in much more close proximity to one another. Remember, action potentials will not be sustained. We can't sustain a depolarization. We have to go through all those steps. Okay, because of that refractory period stuff that we talked about. But if we have a real strong stimulus, we can create action potentials one right after the other in very quick succession. So, for example, a light touch will generate a lower frequency of action potentials, like we have here on the left side of our figure, versus a much firmer touch or pressure will cause a much higher frequency. Okay, that's all the action potential. In addition to this frequency code, a second factor is the number of sensory neurons that will be activated by the stimulus, okay, or recruited. Like we show here with a light initial stimulus, okay, like very lightly pushing in on the skin, that might only stimulate one or two sensory receptors, okay, which ultimately leads to action potentials inward. But if we push much harder in, we stimulate multiple receptors, which ends up with a whole bunch more action potentials being transmitted inward. So the firmer pressure stimulates a larger number of neurons than light touch, and that's how we can distinguish between a very light touch versus a very much more firm touch. Does that make sense? That's pretty cool how that all works. You might have been wondering this since we started to talk about action potentials. Now, another cool concept with regard to sensory receptors is this process called adaptation. We do this all the time. We never really notice it. And that's cool when I say notice it because it's really what it is. Okay. So adaptation is defined as a process in which the generator potential, remember generator potential refers to that initial depolarization, or receptor potential, which is also the depolarization. We're talking about the different types of receptors here. Okay. Decrease in amplitude during a maintained constant stimulus and receptors will vary in how quickly they adapt. Okay. The first classification are the rapidly adapting or phasic receptors, which during the stimulus will generate a action potential burst, but then quickly adapt and sharply reduce their signal transmission even if the stimulus is going to persist. Okay, this happens with like pressure, touch, and smell. Okay. We can show what I'm talking about here on this next figure. Okay. With our letter A, slowly adapting receptor, we're stimulating it from this period from on to this period of off. Okay, versus over here, the stimulus strength is the same for both. Okay, so that's not going to be any different between slowly adapting and rapidly adapting. Okay. What happens is ultimately up here at the top. Okay, and that relates to what happens with the receptor potential. Okay. During the entire time the stimulus is activating the receptor, we're creating receptor potentials that ultimately cause action potentials. Now over time, the action potentials will spread farther apart, so there'll be less coming in because our receptor potential will start to decline. Okay. That's different than what happens with our rapidly adapting, which when we first stimulate it, we create a receptor potential, ultimately causing a couple action potentials. Then, even though our stimulus is still persistent, we are not sending any signal in, and then at the end we do it again. Okay. So rapidly adapting is like what we do with touch and smell. Anybody ever lost their sunglasses when they were stuck on top of their head? Or had their cell phone in their hand and couldn't find it because they were looking all over the room for it? You're touching that whatever, sunglass, cell phone, but do you feel it? Are you consciously aware of it? Not until you start thinking about it, right? Okay. How about when you get dressed? You first put those clothes on, you're going to feel them, right? Okay. How many of you guys were aware that you were wearing clothes just a second ago before I said anything about clothes? No, because it's like they just become part of your body, right? Because what you're doing is not sending signals in to your central nervous system to have perception over that. Is that important to know at all times that we have clothes on? Sure. But is it actually in our brain's capacity important? No. Or the smell of the room. Sometimes students come in here after we've been dissecting something, they say it stinks in here. But then a couple minutes later, do they notice it anymore? No. How about if you jump in a cold pool? It's cold for a couple minutes, then what happens? Does the pool get warmer? No. You decide, is that important to keep sending the signals inward? No. For things like pain, body position, chemical composition of the blood, we're going to continuously send that signal inward. Why? With those things.
You don't want to know when you're in pain.
Yeah, exactly. Okay. Put that hand on the hot stove. Ouch. Okay. If you're able to avoid flexing withdrawing, and you keep your hand on the hot stove and you decide over a second or two, you know what, it's not that important that I send signals in for pain. What's going to happen to you? You're going to get burnt to a crisp, right? Okay. What about chemical composition of the blood? We talked about how like pH is important to our body. What if our pH levels drop and then after a couple minutes of a low pH, we said, you know what, it's not that important. Let's not send the signals inward. Let's not try to fix it. We're not going to survive very long, are we? Okay. These are much more important sensory or senses than these. Not to say that these are not important, but these deal with, really, really important stuff. Does that make sense? Okay. Easy way to demonstrate this is take something like a penny and put it on your forearm. You're going to feel that penny for a little bit, and then all of a sudden it's just going to disappear. Okay, that's stimulating touch, which is a rapidly adapting sensation. We talked about this the other day as well. Some places of our body are really sensitive with regard to these somatic sensations. Remember, somatic sensations are those that arise from sensory receptors in our skin. Teeth of the skin in our subcutaneous layer, mucous membranes, mouth, vagina, and anus, and then our proprioceptors that are located in the muscles, tendons, and joints. And then the inner ear as well, which we're not really going to talk about quite yet. We learned the other day, and we all just decided that our fingers, our hands, and our lips, for example, were really sensitive. Why? Because they are loaded up with sensor receptors, like we're showing over here on the right. A pretty cool test to describe this uneven distribution. It's called a 2-point discrimination test. Okay. These calipers have 2 points on them. They kind of look like a compass. Okay, if you think back to middle school, elementary school, and you had to draw circles. Okay. These calipers have the capacity to change the width, the distance between the 2 points of our calipers. Okay. And when you touch those 2 points, like on our back, they might only end up stimulating one sensory neuron. Okay, because the neurons don't overlap each other like they do in our fingertips, and they're not as loaded in there as like in our fingertips. So what is this person going to feel? They're going to feel 1 point, because they can't distinguish between the two. Okay. Over here, where they're stimulating 2, maybe even 3 sensory neurons, guess what? When you spread them out just a little bit, right away you're going to know that's 2 different points, or 2 points of those 2 different tips. Okay. A pretty cool process. This is called a 2-point discrimination test. Okay. If we had a physical lab in here, we'd actually do this. Okay. You'd find out that your fingertips, your lips, okay, places in your face especially, really sensitive. Okay. Back of your calf, well, your calf is the back, I guess, but your back, your arms, upper arms, places that you're not really coming into contact with a lot of things to physically touch, process them, and have a lot less sensor receptors, and that is illustrated with what we show here on the right side of our graph. Okay. So it's a pretty cool test. Unfortunately, we do it on my lab, so we don't get to really talk about it. You're not talking about it, but actually do it. Excuse me. All right, last thing I want to talk about with the somatic sensations are the different types, and go through some of these in greater detail. But there are 4 modalities or types of somatic sensations: tactile, thermal, proprioceptive, and pain. Okay. Proprioceptive and pain we actually talk a little bit extra about, but tactile and thermal we really don't get too much into, even though we've really talked about them throughout the points of the semester. Tactile sensations include touch, pressure, vibration, itch, and tickle. These are the ones where the receptors are going to be located in the skin or close to it, which are all illustrated on the next page in a figure. Okay. Receptors involved in tactile sensation are called Meissner corpuscles, which are found in the dermal papilla of hairless skin. Meissner corpuscles, or corpuscles of touch, kind of is a newer term for those. They're found in the dermis, which is the deepest layer of our skin, just below our epidermis. Okay. Remember, sometimes the dendrites are covered by a capsule. These are an example of an encapsulated nerve ending. Okay. These Meissner corpuscles sense fine touch, pressure, vibration, and slow vibration. Okay. Hair root plexuses, those are free nerve endings that surround our hair follicles, allowing our hair to be an extension of them and sense things like crude touch. Okay. Merkel discs, or tactile discs, found in the deepest layers of the epidermis. Ruffini corpuscles located in the deep dermis, and Pacinian or lamellated corpuscles, which are found in many places of the body. And here's the Ruffini's, or type 2 cutaneous, lamellated or Pacinian corpuscles, and then those tactile discs or Merkel discs located right there where the epidermis ends and the dermis begins. Okay. You can see various things that they all sense. I'm not really worried about that, but those are all the various tactile sensations. Okay. Now, itch and tickle are stimulated by involve free nerve endings, which cold and warmth also involve, okay, which are the 2 thermal sensations. Cold and warm receptors are both known as thermoreceptors. Free nerve endings having a receptive field about 1 millimeter in diameter on our skin surface. Okay, cold receptors, for whatever reason I don't know, found in the epidermis responding to cold. Warm receptors are down in the dermis responding to warmth. Okay. Pain, I'm going to discuss those in the next few slides. Those are going to involve those free nerve endings, which remember are just the dendrites without a capsule around them. And then proprioceptive sensations are going to involve those that are located in our muscles, our tendons, and our joints. Okay? And they provide us with the ability to know where our head and limbs are located, how they're moving, even if we're not looking at them, so we can walk, type, or dress without using our eyes. Providing us with perception of our body movements called kinesthesia. Collectively, we have 3 proprioceptors which provide us with proprioception. Okay. Muscle spindles, we've talked about those. Those provide us with that stretch reflex. Tendon organs, which are part of that tendon reflex. And I should say joint kinesthetic, not joint, but joint kinesthetic receptors, which do their own thing as well. Let's first talk about pain. Pain is something unfortunate for us, but without it, we would never know if our tissues were being injured, which is the overall original stimulus for pain. Tissue injury or Knox's stimulation. The nerve receptors for this are called nociceptors. We find these actually in all tissues of the body except for our brain. So if you get like a headache, it's not actually your brain that's aching. It's something kind of else. What happens when our tissues get injured is that they release chemicals. These chemicals bind to the receptors on the nociceptors and trigger the actual pain sensation. Most common and most potent pain stimulus is called bradykinin. They're going to activate what we call the first order neurons. The first order neurons are going to be the first in line, transmitting the signal either into the back of the spinal cord or into our brainstem by way of cranial nerves 5, 7, 9, and 10. And from our first order neurons, we're going to synapse with second order neurons, which send signals up through the spinothalamic tract into the medulla, crossover, and then to the thalamus. What was the nickname for the thalamus again? What? Nope. It's your gateway to the cerebral cortex. Because from the thalamus, the third order neurons transmit the signal to the cerebral cortex. Now, how do we know if it's pain coming from our toe versus pain from our finger? We're going to talk about that in a little bit. Okay. Now, something very cool about pain is that we send signals up by way of 2 tracks. Ultimately, the same destination is received, or receiving these signals, are cerebral cortex. Okay. But sometimes the signals travel through the reticular formation in our brainstem. Sometimes they go directly to our thalamus. Okay. If it's going directly to our thalamus by way of that second order neuron, which is our most significant pain pathway, which I would highlight or underline, we call it the spinothalamic tract. This is where most of the somatic pain signals ultimately reaching the cerebral cortex travel. But there are also pain situations where we end up traveling to the limbic system. Now, Sarah, what's the nickname of the limbic system? The emotional brain. Okay. These type of pain signals can trigger visceral, emotional, and behavioral reactions. I'm sure you can all think of a time where you had a pain that triggered some form of emotional response. And we've all experienced it, unfortunately. We can also describe pain many different ways. We have fast pain, we have slow pain, which describes how quickly the signal is traveling inward. Fast pain is a feeling of sharp, localized, stabbing pain right at the time of injury. This type of pain travels by way of myelinated nerve signal or nerve fibers. at speeds of 12 to 30 meters per second. Now this is a yardstick, which is pretty close to a meter stick. Okay. I'm talking 12 to 30 of these in a singular second. Now we're not 12 to 30 meters in length, probably just a couple meters. So that signal is going to get to through your body in a very quick amount of time. Okay. We call this acute, sharp or pricking pain. Very localized and specific to where the pain occurs. Okay. I'm sure you've all stubbed your toe before, right? Okay. When you first stub your toe, it's going to hurt right there in your toe, and it's going to hurt like an SOB. Okay. After a few minutes, what starts to happen though? Feels like your whole foot is on fire, more of a throbbing type pain, right? Okay, it's more generalized. That's slow pain, okay, which is longer lasting, it's dull and diffused. Okay. We travel by way of unmyelinated fibers, still pretty fast, 0.5 to 2 meters per second. So about 1 to 2 of these, maybe. But that's not nearly as fast as what we see here. Okay. We call this pain chronic pain, so like a aching back, okay, burning, even aching or throbbing, okay, which you might feel after a long day of being on your feet. Your dogs are barking, as people like to say. Okay. We can also describe pain based on where it's located. If it's pain coming from our skin, we call that superficial somatic. Superficial is a word that means towards the outside of our body, which your skin is the outside of your body. Deep pain makes sense. It's deep to the skin. Places like our skeletal muscles, joints, tendons, or fascia. And then visceral comes from our viscera, so just like your stomach pain type thing. And viscera, remember, are the internal organs. As I just mentioned on the previous page, fast pain, very precisely localized to the stimulated area. For example, if someone pricks you with a pin, you know exactly which part of the body was stimulated, whereas somatic slow pain is much more diffused, involving larger areas. That's that, your whole foot throbs after a few minutes of having that sharp pain. What's pretty crazy is sometimes we have mixed signals occurring, where visceral pain coming once again from our viscera, is actually interpreted by our brain as pain coming from our skin. We call this phenomenon referred pain. Okay. And I can show you why this happens. Okay. Down here at the bottom of our next page, we can see we have first order neurons coming from the skin and from our heart, which both travel into the back of the spinal cord, and they synapse with the same interneuron, which then travels that signal up to our brain. That second order neuron is going to carry the signal into our brain the same way it would if the signal was originating in our skin or from our viscera. So it might end up being that our brain assumes the pain is coming from our skin instead of where it's actually coming from. So it's kind of like a crossed wire situation. So overall mix-up. And the heart is a great example of this, because our heart, when we have a myocardial infarction, demonstrates this. What is a myocardial infarction? Heart attack. And what's something that people a lot of times will describe as having when they're having a heart attack? Their arm or numb or pain, throbbing, aching, shooting pain, and you can see, especially their left arm, all the way down into their fingers. Yeah. Somebody very smart was able to actually map out these different areas where we would feel deep pain when it's actually, or we'll feel skin pain when it's actually deep pain, visceral pain. Okay. Some of them are pretty wild, like your bladder, actually feeling that down in your legs, lung and diaphragm up in your neck, liver, gallbladder, all which are located right down in here, all the way up in your shoulder and neck. Pretty wild, but can be very helpful for doctors that they know these things, especially in the case of like a heart attack. Somebody has pain in one arm, pain in their chest on that side as well. Boom, they know exactly, probably a heart attack. Serious medical emergency. So if you ever wonder why, now you know. All right. Proprioception is going to be our last of the somatic sensations. After this, we'll get into the special senses. Proprioceptive sensations allow us once again to know where our head and limbs are located and how they are moving, even if we're not looking at them, which enables us to walk, type, or dress without using our eyes. The perception of our body movements is called kinesthesia. The receptors involved are collectively called proprioceptors, and they are known as muscle spindles in our muscles. We've talked about those before. Tendon organs or Golgi tendon organs in our tendons. We've also talked about those before. And then joint kinesthetic receptors, which we haven't really talked about before. Ultimately, these three different types provide us with information on the degree to which muscles are contracted, the amount of tensions placed on tendons, position of our joints. This allows us to perform what's called weight discrimination, ability to assess the weight of an object, which would be especially important if you're trying to lift something up that is super heavy or super light, so you know how much force you need to utilize to perform the task, how much muscular effort is necessary. So let's talk here about the actual types. Once again, we've already talked about the first, muscle spindles. These are found in our skeletal muscles, monitoring changes in the length of skeletal muscles. We talked about them when we talked about the stretch reflex, like your knee jerk. Okay, when we tap on that tendon, the muscle stretches, they stimulate those muscle spindles, which wrap around those muscle fibers. Okay. Now, by adjusting how vigorously a muscle spindle responds to stretching of a skeletal muscle, our brain is actually going to set an overall level of muscle tone, which we've talked about before. Muscle tone is that very slight degree of muscle contraction, even when we are keeping our muscles in a resting state. Over here at the tendon is where we'll find the tendon organs. Like I said, sometimes they're called Golgi tendon organs or GTOs. Involved in tendon reflexes like that reflex that we learned about previously. They cause our muscles to relax when too much tension is placed on them, so they prevent too much damage or any damage to our muscles and tendons. Joint kinesthetic receptors found around the articular capsules, which are what hold our synovial joints together. Synovial joints are our major joints. Could be as complicated as our shoulder or as simple as like the tips of our pinky in the middle of our pinky. Collectively, these respond to pressure, acceleration, and deceleration of joints during movements and can adjust muscles when excessive strain is placed on the joint. Now, I've already been talking about first order, second order, third order neurons, but this is pretty cool to talk about and important as well. Those various somatic sensory sensations, or somatic sensations, as I say, not sensory sensations. Somatic sensations are going to relay information from the receptors that we just described, ultimately landing in our primary somatosensory area of our cortex. Remember, the primary somatosensory area was one of those functional areas that we talked about in our last unit. Okay. The pathways to the cerebral cortex consist of thousands of sets of 3 neurons. First order neurons, which carry the signal from the receptor, or in some cases are the receptor, into the brainstem, which in this figure here we're showing the spinal cord. So going into the back of the spinal cord, and then up into the brainstem, where we have second order neurons that carry the signal up to the thalamus. We learned previously about decussation. Do you guys remember where decussation took place? This was crossing over, took place in the medulla oblongata. We switch sides and then travel to the thalamus, which we know is our gateway to the cortex. And then from there, the third order neurons carry it to the cortex. These somatic sensory impulses ascend to the cortex by way of 3 general pathways. We've talked about these before. Posterior medial lemniscus pathway, anterolateral pathway, and trigeminal thalamic pathway. Now, what is really cool is the primary somatic sensory area in our cortex is mapped out, meaning that there are specific regions within that area of our cortex that signals will travel coming from the various places of our body. Notice how big our hands are and our faces in our figure. This is sometimes known as the homunculus. Okay. Why do you think these places represent really big parts of our figure? Yeah, exactly. These are the most sensitive places on our body, right? Okay. Well, we can stretch out those places so that we can really pinpoint whether that signal is coming from the tip of our finger, the middle of our finger, the back of our finger, the side of our face, the tip of our nose. Places that are a lot less sensitive probably don't make up as big a space. This is cool because this is what allows us to know if the signal is coming from our fingertip or our back of the elbow or our face. So this interprets the signal, which could be pressure here, there, wherever, and enables us to know exactly where it's coming from. Now, we also have this same setup in front of that central sulcus in our precentral gyrus for our primary motor area, which, remember, the primary motor area controls the voluntary skeletal muscle movements of our various body parts. But since this lecture is all about sensation, we're not going to worry about that. What we're going to begin today and finish up on Monday is talking about our 5 special senses. Our first sensation is called olfaction, which is a fancy way to describe smell. Just like the other special senses, it has a very low threshold, which means that it does not require a lot of stimulus to create an action potential. Only a few molecules of certain substances are needed to be present in air to be perceived as an odor. Our olfactory sense organs consist of epithelial support cells and specialized olfactory receptor neurons, which we find in a region of our nose called the olfactory epithelium. Okay, here on the next page, we can show where that is. Okay, in the middle of our figure on the right here, we show a purple shaded region. Just looking at the top of our nasal cavity, we're looking at half of our nasal cavity here, so one side, the left side. We've cut the head in half to look at the inside of it. Located on the inside of that nasal cavity is the olfactory epithelium, which we're zooming in on down here. Okay. Olfactory epithelium is home to 3 cells. Okay. The most important ones are these olfactory receptor cells. These are the actual first order neurons. These are bipolar in structure. Guys, remember bipolar is where the cell body is located on the inside of 2, or in between, I should say, 2 long axons. Here we can see a singular bipolar neuron cell body here, axon on one side, dendrite on the other side. Okay. At the tip of the axons are where we find the actual receptors for our odorants. What we actually smell are called odorants. The molecules and chemicals that we smell stimulate these olfactory hairs or cilia. Okay. These are tiny hairs projecting from the dendrites of the olfactory receptor cells. You can't see them labeled, but they would be these little squiggly lines down here at the bottom of our olfactory epithelium. Okay, now they're going to be covered in mucus, which we know mucus is in our nose, produced by glands called olfactory and Bowman's glands. I'm not really worried about those. Okay. Those are another group of cells that we find in the olfactory epithelium. not really that important to us in this class. The other two cells include supporting cells, which provide support nourishment to the olfactory epithelial cells. They also insulate electrical signals, okay, and detoxify chemicals that they come into contact with. The third type of cells in the olfactory epithelium are called basal stem cells, which replace the worn out and dying olfactory receptor cells. Okay. Olfactory receptor cells don't have a super long lifespan. Okay. Now let's talk more about the olfactory receptor cells. Each side of our nose, we have 40 bundles of these olfactory receptor cells that extend down through a hole, or holes, I should say, in this plate of bone that is at the top or is the roof of our nasal cavity. This plate of bone comes from our ethmoid bone. It's known as the cribriform plate. Some of you guys are taking anatomy now. This should be fairly familiar if you've taken anatomy in the past. Hopefully that also rings a bell. Okay. They extend down through these holes, as we can see down here. And collectively, these 40 or so bundles of axons form our right and left olfactory cranial nerves. Remember, olfactory cranial nerves were the first pair. Okay. Now, these olfactory cranial nerves terminate in the brain in paired masses of gray matter, which we call the olfactory bulbs, which we can see one is labeled here in our middle figure. This olfactory bulb is just a swelling located at the end of our olfactory cranial nerves. And it sits right above that ethmoid bone. Okay. Inside of our olfactory bulb is where those olfactory receptor cells synapse with our next set of cells. Okay. The next set of cells are called olfactory bulb neurons. They're labeled here as mitral cells, but they are collectively going to form our olfactory tract. Okay. And that's how they exit the olfactory bulb and away from the nose towards our brain, as you can see there in the top part of our figure. Okay. With our yellow line running towards our cerebral cortex. Now, some of the axons will project to that primary olfactory area in our temporal lobes to allow us to have conscious awareness of our smells. Some of those signals will travel to our limbic system and hypothalamus. Once again, Sarah, what was our limbic system? Emotion brain. You ever think of an instance where you have a smell that triggers an emotional or some form of memory evoked response? Perfumes and colognes, deodorants. These are things that people wear and smell good, and then sometimes you associate people with those smells. Okay? Or like a loved one that's off at basic training. You still have their favorite hoodie, and you can smell them, and you can trigger all sorts of emotional responses or memories. So you might not think about how amazing smell is. Maybe now you do. Now, in order for this to all occur, we have to first stimulate those olfactory receptors. And that means we have to bind odorant to the olfactory hairs. What we are doing in the figure here on the right is zooming in at one little segment of our olfactory hair. Here's the plasma membrane, and it's loaded with various proteins. Okay. Smell involves what we call a second messenger system, which I wrote up here on the board. Second messenger system means that there are two messengers involved. The first messenger is literally the odorant, which in this case, you see step one, the odorant is going to bind to a receptor protein located on that plasma membrane. That then activates what we call a G protein on the inside of the cell, which then activates an enzyme called adenylated cyclase, which converts ATP to cyclic AMP. This is our second messenger right in here. Cyclic AMP then triggers the opening of cations. Things like sodium can rush in. Guess what happens when sodium rushes in? We depolarize, and if enough sodium rushes in, guess what happens? We create an action potential or nerve impulse in those olfactory receptor cells. They then transmit that signal inward to the olfactory bulbs, then to the olfactory tract, and then ultimately the primary olfactory area of our cerebral cortex. Now, are you going to need to know all this stuff? No. But I definitely want you to know that smell involves what's called a second messenger system. And the cranial nerve involved in this, cranial nerves 1. Now, our next sensation is taste. And the great thing about taste is it's actually really, really similar to smell. Both of those are chemical sensations. We had odorants for smell. Now we have what we call tastants for taste. The fancy way to describe taste is gestation. And we use our tongue, and specifically the taste buds that are located in our tongue for taste. Okay. Now we have taste buds kind of in our soft palate and pharynx as well, but most of them are found in our tongue. Okay. Found in elevations on our tongue called lingual papilla. There are 4 different types of lingual papilla. Filiform, which really aren't important to us for the sense of taste, because they have no taste buds, without taste buds. They do have tactile receptors, so they do provide us with, the sense of touch of our food. Okay. They also increase friction between the tongue and food to help it move in the oral cavity. The second type really aren't important to us, especially since we're all adults, because they degenerate early in childhood. The third and fourth are important to us, especially the fourth. Fungiform, there are about 5 taste buds located in each of those, but the majority of them form this kind of inverted V in the back of our tongue called valate or circumvallate. And if you zoom in at one of them, you'll find inside our taste buds, which is what we're showing here on our figure on the right. Now, just like in our nose, we have 3 cells. At least in our olfactory epithelium, we have 3 cells. We have the actual receptor cells. We call these taste cells or gustatory receptor cells. These are the yellowish orange shaded cells. They're banana shaped. Okay. And at the tip of them, just like at the tip of our olfactory receptor cells, we have little hair-like structures. We call these taste hairs or taste microvilli or gustatory microvilli. We just have them labeled here as microvilli. Okay. And those are the actual receptor sites for the food molecules. Okay. At their base, they synapse with first-order neurons. Okay. So they're a little bit different because these are examples of separate cells. They're not the actual neurons, but they synapse with the neurons. Okay. And we'll talk about that a little bit more here in a second. Okay. Basal cells are these kind of purplish ones, which will then develop into the supporting cells. Okay. And they also contain microvilli because they will end up becoming the receptor cells. So these cells become these cells, which then become these cells. So that's kind of cool. Ultimately, we only actually have 5 taste sensations, primary taste sensations. And in order to process these taste sensations, we first must stimulate those taste hairs or taste microvilli. This involves our saliva, actually. Okay. Saliva allows those tastant molecules to dissolve and then penetrate into these taste buds, which are kind of found in these little pits in our tongue and those little papilla. Okay. A cool way to actually prevent this is actually just drying your tongue. This is something I would do if we had a physical lab. I'd have you guys all dry your tongue and then sprinkle some sugar on your tongue. Hold your tongue out. You're not going to taste that sugar for probably a minute or so. Okay. Versus if your tongue was wet, you would drop that sugar on there, you're going to taste that sweet sensation right off the bat. Okay. Sweet, by the way, is one of our 5 primary taste sensations, produced by many organic compounds, particularly sugars. Salty is our other one there, first listed actually, produced by metal ions like sodium and potassium. Sour, sensation associated with acids. Bitter, produced by alkaloids, designed to cause an animal to reject the food, as in it's spoiled or diseased. And then umami, which is a meaty taste produced by amino acids. Meaty or savory, it's a Japanese word, umami. Now, you guys would all agree that there are plenty more tastes out there than just those 5, right? Everything is just a combination of those 5. Now, down at the bottom here, we can show how taste is a little bit different than smell. Remember, smell involved those bipolar olfactory receptor cells. Taste involves separate cells, which we can see a separate cell here, which is called a gustatory receptor cell, or just a taste cell, and then another one over here. Now, those 5 primary taste sensations can either directly cause depolarization or have to utilize a second messenger system like what we did with smell. For example, something salty is going to probably have sodium in it. Something like salt, which is NaCl, going to have salt or Na in it, Well, if Na can go in through a sodium channel, guess what's going to happen to this cell? It's going to depolarize, right? That ultimately causes the release of neurotransmitters that then synapse with our first order neuron. Okay. Same thing for hydrogen ion, like in a sour taste. Over here on the right, the ultimate result is the same, release of neurotransmitters, except we get there a little bit trickier because we have to activate a second messenger system. Now, once the first order neurons are stimulated, we're going to do the same thing that we saw earlier. We're going to transmit that signal inward to our brain. This time, though, we actually have 3 different pairs of cranial nerves because our tongue is innervated at different points by those 3 different cranial nerves. For example, the tip of our tongue and most of the tongue innervated by our 2 facial nerves. And we're only showing 1, but remember these are all in pairs. Okay, towards the back, what we'll have is the glossopharyngeal nerve, and then the very back, covering our epiglottis, is the vagus nerve. Okay, and that will be on both sides, so both pairs. Okay. Those 3 cranial nerves are going to propagate the impulses to the medulla, and then on to the limbic system, and then to the thalamus, hypothalamus, or limbic system, just like we saw with smell. If it's going to the limbic system, we're talking about stimulating emotional and memory-evoked responses. Okay. Think back to a time when you were with your boyfriend and you ate something and you tasted it again and it brings back that wonderful memory. But we also have signals that then travel to our cortex from the thalamus. That cortex will be the primary gustatory area, which is found in our parietal lobe. Okay. And that gives us our conscious perception of taste. Any questions on taste? All right, we will pick up with audition or hearing on Monday.